Hello, everyone. This is Dr. Richard Fleming, and today is Wednesday, the 27th of January, 2021. During our last couple videos, we looked at the emergency use authorization documents filed with the FDA for the Pfizer and Moderna vaccines. In those documents, we reviewed information regarding the efficacy and the absence of efficacy noted in those EUA documents. As of the National Clinical Trials website that I looked at yesterday, the trials for both Pfizer and Moderna appear to be active. However, they're not recruiting for further patients to investigate those vaccines at this time. The purpose of today's video is to review three basic safety concerns for these vaccines. The first is something called antibody-dependent enhancement. The second has to do with prions and their role in the spike protein and the potential risks that they pose to people receiving the vaccines. And the third has to do with the ability of the genetic information, the mRNA of the coronavirus in the vaccines, to insert itself into the DNA of the human nucleus, our cells, and become a part of our permanent genetic code by inserting into our DNA. To do that, I'd like to review several documents with you today. And so I'm going to share the screen so that you can see exactly which documents those are. And I want you to know that you can find these documents available on this website where we're posting information, www.flemingmethod.com. This is the first page. And if you go to the documentation tab over here and click on that, you will see information that we're going to be covering, as well as the actual documents available that we are reviewing in this video. Let's begin with emergency use authorization and what it really means. This recommendation document put out by the FDA is dated October 2020, and it seems to be the most up-to-date document. By the U.S. Department of Health and Human Services, Food and Drug Administration, and the Center for Biologics Evaluation and Research from the federal government. And as we scroll through this document, we're going to see that it specifically notes that in general, FDA guidance documents, which this is, including this guidance, do not establish legally enforceable responsibilities. Instead, guidances describe the agency's current thinking on a topic and should be viewed only as recommendations, unless specific regulatory or statutory requirements are cited. Those would be laws passed by the elected officials in Congress. The use of the word should in agency guidance means that something is suggested or recommended, but not required. There are four primary components or criteria for an emergency use authorization, as put together by the Secretary of Health and Human Services, determined to be in the interest of the public health in an emergency. These four justifying the authorization of emergency use. Based upon this declaration and determination, FDA may issue an FDA or an EUA, and the four items are: the result has to do with a serious or life-threatening disease or condition, of which I think we can agree that SARS-CoV-2 poses a life-threatening disease or condition, given the number of deaths. Based on the totality of the evidence, is the product, in this case the vaccines, effective? You could also raise this question for the PCR test, whether it's actually effective. But we're staying with vaccines right now. The third is that the benefits of the vaccines actually outweigh the known or potential risks. And that brings us to our topic today. And finally, The requirement for an emergency use authorization of a vaccine or a test or drug requires that the FDA has not previously provided that there's an adequate, approved, or alternative treatment to the product for diagnosing, preventing, or treating the disease or condition. In the case of investigational vaccines being developed for the prevention of COVID-19, any assessment regarding an emergency use authorization will be made on a case-by-case basis, considering the target population, of which, as we've seen from the EUA documents, there's a lot of missing information from a variety of target informations. In fact, yesterday when I was looking at the side effects, reported adverse effects on the vaccines, one of the things that caught my eye was a child less than 3 years of age reported to have adverse event to the vaccines. It boggles my mind to think that somebody actually vaccinated a child less than 3 years of age using an experimental vaccine where it's been noted by the FDA and the emergency use authorizations that there's no data in people below the age of 16. When we look at the Pfizer vaccine, one of the specific statements that they made in the emergency use authorization was that there is no data to indicate that there is a vaccine-enhanced disease associated with the vaccine. As I mentioned previously, this document, this EUA, was filed on the 10th of December. And we're going to see shortly that the Osaka paper shows adverse enhanced infectivity resulting from the virus on the 18th, 8 days after this was filed. The Moderna paper notes the same thing, and this was filed on the 17th of December, 1 day before the Osaka document was published. The Osaka paper that we're going to get to in just a moment is not the first paper to show that vaccines to SARS viruses produce antibody-dependent enhancement, ADE. In fact, vaccine efforts to the first SARS virus, originally in 2011, with the virus that was present in 2002 to 2004, has the following abstract entitled Anti-Severe Acute Respiratory Syndrome Coronavirus Spike Antibodies Trigger Infection of Human Immune Cells through very specific pathways. Public health measures successfully contained outbreaks of the severe acute respiratory syndrome coronavirus infection, which I now call SARS-CoV-1, since we now have SARS-CoV-2. However, the precursor, the SARS-CoV virus, remains in its natural bat reservoir, and reemergence of a human-adapted SARS-like coronavirus remains a plausible public health concern. In 2011, vaccination is a major strategy for containing resurgence of SARS in humans, and a number of vaccine candidates have been tested in experimental animal models. These authors previously reported that antibody elicited by this vaccine candidate based on the full-length spike protein potentiated infection of our immune cells, our B cells, despite a neutralizing antibody showing some protection. So there was a problem already noted in 2011. The observations prompted us to investigate the mechanisms of underlying antibody-dependent enhancement of the SARS-CoV infection in vitro. We demonstrated here that anti-spike immune serum, while inhibiting the virus, potentiated infection of the virus. It went on to comment that this illustrates the potential pitfalls associated with immunizing against the virus. Since the introduction of SARS-CoV-2, there have been papers showing this antibody-dependent enhancement. And it began with mice and non-human primates. monkeys. When we go down here and look at the abstract of this paper, it specifically states that neutralizing antibodies to the SARS-CoV-2 virus, protecting against coronavirus disease, makes them a focus of vaccine design. A safety concern regarding the SARS-CoV-2 antibodies is whether they mediate disease enhancement. Here we isolated the neutralizing antibodies against the receptor binding domain. That's the part that attaches to the ACE2 receptor, and the N-terminal domain, another part of the spike protein. And they looked at it in individuals who had recovered from infection, the convalescent plasma, or a history of infection. Then they looked at it with cryo-electron microscopy to look at the antibodies to both the receptor binding domain and the N-terminal domain. What they discovered was that there were antibody-dependent enhancement, making the virus more infective, enhancing viral infection in vitro, outside of the people. But they went further to look at the animals and discovered that, in fact, if you look at the ratios of how many antibodies are made to the receptor binding domain versus how many are made to the N-terminal domain of the spike protein, you see that the more antibodies to the N-terminal domain that are made, the more severely ill the patient is, and the more likely they are to die. And in fact, in animals that were thought to be non-infectious, but when they looked at their blood, 3% of the monkeys that were looked at actually had more N-terminal domain antibodies present, and transfusing that blood into somebody else would potentiate more infection from the SARS-CoV-2 virus. On the 18th of December, physicians and research scientists in Osaka, Japan, published a paper that says an infectivity enhancing site on the SARS-CoV-2 spike protein is targeted by COVID-19 patient antibodies. This is the same N-terminal domain concern seen with the animal models. And here they state, the SARS-CoV-2 infection causes severe symptoms in a subset of patients, suggesting the presence of certain unknown risk factors. Although antibodies against the receptor binding domain of the SARS-CoV-2 spike have been shown to prevent the SARS-CoV-2 infection, the effective antibodies against the other spike domains are largely unknown. Here we screened a series of anti-spike monoclonal antibodies from COVID-19 patients and found that some of the antibodies against the N-terminal domain dramatically enhanced the binding capacity of the spike 2 protein to ACE2, and thus increased the SARS-CoV-2 infectivity. Surprisingly, mutational analysis revealed that all the infectivity-enhancing antibodies recognized a specific site on the surface of the N-terminal domain. The antibodies against the infectivity-enhancing site were detected in all samples of hospitalized COVID-19 patients in the study. And it was the ratio that differed among the outcomes of the patients. Furthermore, the antibodies against the infectivity-enhancing site were detected in 3 out of 48 uninfected donors. These findings suggest that the production of antibodies against SARS-CoV-2 infectivity-enhancing site could be considered as a possible exacerbating factor for COVID-19, and that a spike protein lacking such antibody epitopes may be required for a safe vaccine development, especially for individuals with pre-existing antibodies. In other words, people that have been infected with SARS-CoV-2. The animal model paper and the Osaka paper specifically demonstrate that there is evidence of a vaccine enhancement of disease for both vaccines that carry the entire spike protein like the Pfizer and Moderna vaccines do. The Osaka paper is very clear. The animal model is very clear. There are antibodies made to the spike protein which are present in the vaccines that increases the ability, the infectivity of SARS-CoV-2. We also now know that a second problem has been present, and this was originally noted in March of last year. Something that Pfizer and Moderna should have been aware of, and something that the FDA should have raised questions about. SARS-CoV-2 prion-like domains in the spike proteins enable a higher affinity for the ACE2 receptor. These are prion-like domains. Already noted back in March of last year. Currently, the world is struggling with the coronavirus 2019 pandemic caused by the severe acute respiratory syndrome coronavirus 2. Prion-like domains are critical for the virulence. These prion-like components of the spike protein that is coded for in the vaccines and the development of therapeutic targets. However, the prion-like domains have not been analyzed. These SARS-CoV-2 is the only coronavirus with these prion-like domains. One of the reasons that we can tell that it is so much different than anything else that's been seen, indicating gain of function. This results in a 10 to 20-fold higher affinity for the SARS-CoV-2 virus than any other virus, any other coronavirus that's out there. And these prion-like domains have been identified in the SARS-CoV-2 receptor binding region of the spike protein. That's in the virus vaccines. These viral-like domains represent a new regulation for viral assembly. And it's what's seen in humans with Alzheimer's, Parkinson's disease, diabetes, and a number of human diseases. These malfolded proteins that are present, these viral prions that are present on the spike protein that we're coding for in the vaccines. Papers were published back in 2018 showing these prion-like areas producing amyloid precursors that are involved in Alzheimer's disease and other neurologic diseases. And for those of you who may not be familiar with prions or amyloid diseases, they produce a variety of problems. They are abnormally folded proteins. They're involved in Alzheimer's disease, Huntington's disease, Parkinson's disease, and the very specific things that you should associate with prion diseases are scrapies found with sheep, bovine spongiform encephalopathy, BSE or mad cow disease, Kuru, which is seen with the eating of monkey brains, Creutzfeldt-Jakob disease, which I've actually seen and treated or attempted to treat. It's a fatal disease, GSS and other familial diseases. They're associated with certain genetic defects or infections by infected tissue or tissue derivatives with these prions, producing these amyloid plaques in the brains, producing the infections. So 2 risk factors that have not been taken into account from the EUAs so far have to do with antibody-dependent enhancement, now shown to be a fact for antibodies made to the spike protein, and the prions that are present in the spike protein, now shown by multiple published papers. And finally, SARS-CoV-2 RNA reverse transcribed and integrated into the human genome. Now, many people have speculated and talked about whether these vaccines or this virus could have been done for the express purpose of getting into human DNA. And for many months, I simply stated that it's an interesting concept, but there's no published data to show that that happens. So before one could entertain the idea that perhaps something is being included in this SARS-CoV-2 virus or in the vaccines, you would have to prove to me that in fact there was a way for the SARS-CoV-2, either the virus itself or the vaccines, to get into human DNA. And lo and behold, in December of 2020, the 13th, which is 3 days after the Pfizer EUA and 4 days before the Moderna EUA was filed and approved or filed, this paper comes out showing that the mRNA of the virus and the vaccine carries the necessary genetic information to cause the reverse transcription, the placing of the viral RNA into human DNA. Prolonged SARS-CoV-2 RNA shedding and recurrence of PCR positive tests have been widely reported in patients after recovery. Yet, these patients most commonly are non-infectious. Here in this paper, the authors investigated the possibility that the SARS-CoV-2 RNA can be reverse transcribed and integrated, put into the human genome, our DNA, and that transcription of the integrated sequences might account for the PCR positive tests. In other words, these authors looked at whether the virus being in our cells could insert itself into our DNA and then later reappear, re-express itself after we thought people were recovered. And if you, as you'll note on this site where we talk about SARS-CoV-2, almost a third of recovered COVID patients returned to the hospital in 5 months, with 1 in 8 of those patients dying. These patients clearly are representing even after they were thought to have recovered. In support of this hypothesis, this test question, we found chimeric transcripts. A chimer is a merging of more than one species, the virus with the human. We found chimeric transcripts consisting of viral-fused to cellular sequences, our cells, in published data sets of SARS-CoV-2 infected culture cells and primary cells of patients, not just a test tube, but patients, consistent with the transcription of the viral sequences integrated into the genome. And as you look at these documents, as you look at this image, you will see evidence that the mRNA of the virus, which is shown in red here, and the human DNA shown in blue in the nucleus, that the virus has entered the nucleus and here has integrated, put itself into human DNA. This is no longer a theory or a question. This is now a scientifically proven fact. To experimentally corroborate the possibility of viral retrointegration, we describe evidence that SARS-CoV-2 RNAs can be reverse transcribed in human cells by reverse transcriptase, the same thing HIV has. And remember, one of the gain of functions of this virus, in addition to the PRRA insert that is not seen on any other coronavirus, but is seen with HIV, glycoprotein 120, and cobratoxin and rabies that was inserted into this virus. There's also the HIV pseudovirus glycoprotein 120, which has reverse transcriptase properties. But if that wasn't bad enough, our human genetic material, 17% of it, codes for this reverse transcription, the ability to take something and put it into our DNA if it's the right stuff, in what's called reverse transcriptase from the line 1 elements. And as you're going to see in this paper, that means the long interspersed nuclear elements in human DNA that accounts for 17% of our DNA. So we have within our DNA, if the right virus attacks or the right genetic material from a vaccine attacks, to put that genetic material into our cells and pass it on from cell to cell to cell, keeping alive what's been presented to us either by the virus or the vaccine. Our human endogenous, that which occurs naturally in us, our line 1 expression, was induced by the SARS-CoV-2 infection or the cytokine responses that are produced in the inflammation and thrombosis, or the inflammothrombotic response, suggesting a molecular mechanism for the SARS-CoV-2 retrointegration into patients. A novel feature of the SARS-CoV-2 infection may explain why patients continue to produce viral RNA after recovery, which means that whether they got it by the exposure to another person or the vaccine, this can now be transmitted to other people, and giving it to people by the vaccine only makes it possible for people to be asymptomatic and spread it to other individuals. Continuous or recurrent positive PCR tests have been reported in patients weeks and months after recovery from an initial infection. A bona fide reinfection, although a bona fide reinfection of SARS after recovery has been reported, cohort-based studies, similar studies of similar people with strict quarantine or subjects recovered, suggest a repositive cases was not caused by reinfection of emergence of the original infection, because it was fused in our DNA to then reinfect us. Furthermore, no replication competent virus was isolated or spread from PCR positive patients. The cause for such prolonged and recurrent viral DNA or RNA production is unknown. A positive stranded RNA virus, the SARS-CoV-2 and other similar beta viruses, such as SARS-CoV-1 and MERS, employed RNA-dependent RNA polymerase to replicate their RNA and transcribe into the subgenomic RNAs. One possibility is that this SARS-CoV-2 virus could be reverse transcribed and integrated into the human genome and transcribed, and the integrated DNA copies could be responsible for positive PCR tests because it's being re-expressed. It's inserting itself into our DNA, and then our DNA makes new DNA with cells, and the SARS-CoV-2 can reemerge coming out of our very DNA. For example, the reverse transcribed endogenous, producing these APP fragments into the genome of our neurons themselves. And this is where it becomes an additional problem when you see the prions, because of all the diseases that are caused in the brain. And amyloid is also a disease in the heart. Amyloidosis, amyloid heart disease, produces a weakened heart in people who have it. So these prions producing these amyloid-like diseases not only will interfere with the brain of people, but the heart of people, simply by getting exposed to the virus. And the vaccines are promoting even more of these prions and this SARS-CoV-2 spike protein with the reverse transcriptase. We've known this in cardiology for years, because this paper looked at the ability to have this long interspersed nuclear element to result in a reverse transcriptase in platelets. Cardiologists like myself have looked at platelets for a long time, but we also know that this is present in 17% of all cells, including, as we know from the HIV virus, this occurs in our CD4 cells, the very cells called to respond to these infections in our immune system, make it possible for this virus to get into our CD4 helper cells, are T cells responsible for the initial response to the virus and integrate itself into our very cells, making this virus persistent with long-term sequelae. And introducing this vaccine introduces even more than could possibly be seen in the natural setting. Let me show this one here. This is the sequence that you need to be aware of when you're looking at what happens naturally versus these vaccines. I'm going to enlarge this, and then I'm going to take us just to the top of it to begin with, and we're going to walk through this, but there is more information available for this, again, on this website, on FlemingMethod.com, where we talk about not only this ability to measure what's really going on, but in the Documents tab where we are presenting for you to read at your own pace with the documents that I'm showing you today, exactly what happens and the difference between the naturally occurring virus and what's happening with these vaccines. And so let's go back to this enlarged version of this image to make it easier for everybody to see. And we will begin by walking through. This image shows the antibody pathways that occur and how they're impacting the coronavirus disease. Now, there's two pathways we're looking at here. One is what happens when this virus is transmitted from person to person. It's hard to call this the natural pathway because the evidence demonstrates that there has been gain of function provided to this virus through the PRRA insert, through the HIV pseudovirus glycoprotein 1 insert, and we're seeing prions in this spike protein that we've never seen with any other coronavirus. Three things showing up that have never existed before, suddenly showing up after we have, as shown in prior videos, admission of gain of function research occurring funded by the federal government, by the US federal government, with work done through EcoHealth and Peter Daszak, Ralph Barrick at the University of North Carolina, and Dr. Shizheng Li at the Wuhan Virology Institute. So this SARS-CoV-2 virus that is spread from person to person in the air has spike proteins, and you have all come to think of this as a red triangle. And this virus spike protein will naturally occur and attach itself to the ACE receptor of a cell. But there's only so many ACE receptors to any given cell. These are the doorways, the paths into our cells for the virus. This virus, by adding the PR insert, is particularly attuned to attaching to our ACE receptors, increasing its infectivity by 10 to 20 times, as we saw in those prior documents. When it's floating around our bloodstream, macrophages, our helpers, our T1 helper cells, recognize this and are responsible for showing these spike proteins. These are T cells that generate a response in about 3 to 5 days. And then they present this spike protein to the beta cells, our B cells in our body, which are responsible for making antibodies that will normally do it in about 7 to 10 days. So that's why many people recover from these viruses at about 3 to 5 days, because the T cell response is enough. But other people take longer to recover, and they make antibodies. But remember, the papers have shown us that you not only make antibodies to the regional binding domain of the spike protein, but you also make antibodies to the N-terminal domain, increasing the ability of this virus to infect people. So many people don't get better because when they make their antibodies at 7 to 10 days, not only do they make the ones that attack the spike protein which is good, but they also are making antibodies to the N-terminal domain, as we'll show you farther down in this image, enhancing the infectivity of the virus. When people ask, will the antibodies that we make, either because of seeing the virus pass from person to person, quote, naturally, or antibodies that are made in response to the vaccines, well, the more the mutated spike proteins occur, the less the antibody will recognize it. So for now, with the original looking, antibodies will attack the regional binding domain, but with more and more mutations, it's just a matter of time before these antibodies can't recognize it. They see it, but they can't attach, they can't do anything to it, and so they will be useless over time. That's the naturally occurring. But remember, only so much of this virus can get into your cells to replicate and come back out to start the reaction of the T cell macrophages, finding the virus, showing it to the B cells to make antibodies to attack. Limited number of doorways. And so for many people, the ones who've had problems have been those who fall into extremes. Those that are immune naive, the very young, have diabetes, so they have an impaired immune system, have an immune deficiency disease, so they're not geared up and they're not working very well. Or the people with all the hyperinflammatory diseases, as I presented originally in 1994 at American Heart, and again in 1995 at American Heart, and published in a textbook, a cardiology textbook in 1999, and then showed with research studies that we published from 2000 to 2003. And as we talked about on 2020 and 2004, this inflammation, these inflammothrombotic diseases are the result of a variety of problems, including bacteria and viruses that cause blood clots and inflammation, causing heart attacks and strokes and blood clots to the lungs and kidney failure and cancer and other diseases. That when this inflammothrombotic occurs in people with these, what you now have called comorbidities, all these health problems that I presented in the theories to explain this, they're hyperactive to begin with. So for them, they already have a hyperactive response. But for the normal group, that middle-aged group, the 20s to 65-year-olds that we didn't see as many deaths in, they were responding to a normal amount of the virus being able to enter our cells through the ACE receptors. And so only a standard amount of the production of the virus is being made, something that people without these diseases could respond normally to and address the virus and recover from. And that's why we saw it. But what do these viral vaccines do? Well, these viral vaccines place the genetic information to make the spike protein inside of a lipid layer. And inside of that lipid layer, with the information coding, the mRNA, to tell it to make the spike protein, because it's in a lipid layer, it's no longer dependent upon the ACE receptor. This is a new gain of function that we have given for the virus by making these vaccines. And this lipid layer, this coating around here, then merges with our cells, not just at the ACE2 receptors, but everywhere else, everywhere else where that viral genetic information can come into our cells and give the instruction to make more spike protein to then load up the surface of our cells with more spike protein than could possibly be made by naturally coming through the ACE2 receptor pathway. And that is going to result in all the people that have normal functioning immune systems having an increased immuno response with more inflammation and more blood clotting, more inflammothrombotic response. So the vaccine produces more viral spike protein on the cell wall potential than could possibly occur naturally because it's flooding the cell with more mRNA for the spike protein than could ever possibly get into the ACE receptors. And that's inducing an inflammothrombotic response. And if you look at the age ranges of people having adverse reactions to these vaccines, you will see by and large the vast majority of them are in that age range that we thought were protected from having a problem, the 20s to the 40s. And as I said, shockingly, somebody gave this to a less than 3-year-old, the vaccine, and had an adverse response. And the deaths that are occurring are not only mounting up in the elderly, but across all age ranges, because it's overwhelming and causing a massive immune response. We'd like to see a balanced immune response, not a massive one. And then you have to remember that with reverse transcriptase, that third factor that we saw, that third risk and concern, allows the mRNA of the viral vaccine, of the virus and its spike protein, to insert itself, to get into our nucleus and combine with our genetic DNA, the RNA of the virus, to reverse transcribe. You've heard this before. This is what makes HIV so dangerous. And we don't have a vaccine to HIV. And there are, as you saw, some reasons why already. This shown here, as I already provided for you earlier in the presentation, this red dot shows the mRNA of the virus getting into our nucleus. And the more you overwhelm our cells with more and more mRNA through this vaccine, the more of this there will be to insert itself into our genetic code and be passed every time the cell divides, allowing the cell to then re-express the virus later on, not to mention whatever other potential harm has been introduced into these vaccines. Because remember, we're taking Pfizer and Moderna and the FDA's word that the only thing in these vaccines are the mRNA of the virus. And we know for a fact that without more than just the spike protein mRNA, our cells won't know to make the spike protein. It also takes the mRNA of the open reading frame to make the enzymes for our cells to make the spike protein. So there's more in these vaccines than just the mRNA of the spike protein. When these spike proteins are made, whether it's a naturally attaching ACE receptor or one made now in response to the vaccine, there are these additional problems. There is the HIV pseudovirus insert. There is the PRRA insert, the proline, arginine, alanine. These are amino acids insert that are not found in any other coronavirus ever seen. They're found in HIV. They're found in cobra toxin. They're found in rabies. And we know, thanks to the videos that I've shared with you previously, that Ralph Barrick at the University of North Carolina has admitted, as has Dr. Sheng Li at the Wuhan Institute of Virology, that they work together to make these chimers, the backbone of one virus with something else from another virus, and put them together to make a mutant called a chimer in medicine or in science, or a Frankenstein in lay terms, perhaps. The same thing, combining body parts of more than one to make something new. This PRRA increases the infectivity. The HIV glycoprotein 120 increases the infectivity. The prion insert, noted here, increases not only the infectivity, but then as a result causes these amyloid plaques that causes heart damage and causes brain damage and causes the neuromuscular disorders and the shaking and the twitching and memory problems and other problems with recognizing how severe the disease is, making you asymptomatic so that you can spread it to other people without knowing how severely ill you are. And the regional binding domain antibodies that are made may be helpful, but the binding antibodies to the N-terminal domain change the structure. In this area here, my spike protein, which is put here for me because as a scientist physician, this is what this spike protein looks like. It's not a red triangle, but a red triangle is a way to think about it. This is the regional binding domain area. This is the area of the N-terminal domain. The glycoprotein 120 HIV insert sits in here, and this shows the flexibility of this spike protein as it hunts and searches for the ACE receptors. And so all of this gets changed when you have an antibody that attaches to the N-terminal domain. It changes what this looks like, which affects the rest of this structure, opening up the regional binding domain to make it more infectious. So there is evidence of this vaccine enhancing the disease by producing antibodies to the N-terminal domain, allowing it to increase the infectivity, allowing it to worsen the disease and to make more of the virus available to be spread. The prion-like domains that are in here increases the infectivity of the virus, and it's included in the vaccines if the vaccines, as they say, include the genetic code for the spike protein. And it includes the HIV insert, and it includes the PRRA, furin site cleavage, which is what this is called scientifically, seen with cobra toxin, with rabies, and with HIV. These are all referenced, documents 7 through 9, 4 to 6, 10 to 11, and back up here for documents 1 to 3 on the website where we talk about it. And you will see these are numbered in the in the new site. Let's take a quick look and see if we have that updated yet. And lo and behold, they're now numbered with the sequences, so you can see 1, 2, 3. You see the updated figure, 4, 5, 6. Down here, 7, 8, 9, explaining the prions, 10, 11, 12, explaining the reverse transcriptase of this virus inserting here. And remember, the vaccines carry more of this mRNA for insertion into our genetic code than could possibly get through to these ACE2 receptors. The EUA documents, as we've been talking about, have already shown a lack of demonstration of the efficacy of these vaccines. Age groups, pregnant people, elderly individuals, immune compromised, the very young. These vaccine trials focused on healthy, young to middle-aged people. They promise to provide updated information, and yet clinical trials show that they currently aren't even recruiting for these studies anymore. They may not need to. With the number of people being vaccinated, I think they're going to have sufficient numbers of people to see what the consequences of their vaccine trials are. But the risks of these vaccines, that they report not having a vaccine-enhanced disease, antibody-dependent enhancement, clearly isn't correct. There are statements that there's no evidence of this virus or this vaccine and this genetic code possibly having any effect on being inserted into our DNA has been shown to be wrong. And the issue of the prions, not even addressed. That would be under their, are there any other concerns? For the FDA to do due diligence on these vaccines requires that they address not only the lack of efficacy shown in these EU documents and raise the questions about why the vaccine trials are no longer recruiting, but they also need to address the risks that we've seen with antibody-dependent enhancement from the vaccines, with the prions, and the damage that it will do to the brain and the heart and other body organ tissues. And with the reverse transcriptase data that shows now that these vaccines and this virus can insert itself into human genetic material. We've left the era of if, to it does. The questions of these risks and the absence of efficacy seriously needs to be raised given the criteria of the EUA by the FDA that the risks are minimal and the efficacy is great and there are no other treatments, raising the questions of why aren't the other treatments being recognized. If other treatments were recognized, then there would be no use for emergency use authorization, and these vaccines would not be approved because they're not approved by the Food and Drug Administration to be used under licensing to be vaccinated people, only under an emergency use authorization. Recognize other treatments as working. Recognize that there is not efficacy really shown, that there's great questions about whether these vaccines provide an effective treatment for the virus. Recognizing that there are serious risks not recognized or discussed in the EUA documents. The FDA has a responsibility to take up these questions. There should be a moratorium on the use of these vaccines. I know people are concerned about getting SARS-CoV-2. Let me assure you, given the things that you've just seen, there are legitimate concerns that you should have, but the vaccines don't eliminate those concerns. They augment, they increase those concerns because we are now passing greater quantities of this genetic code of this virus and putting it into people on a daily basis than we've ever seen before with the respiratory spread. This is a reason to be extremely concerned, ladies and gentlemen. This is a reason for you to spread this video to everybody you know, to raise questions. I'm only presenting the scientific evidence here. It is the responsibility of the FDA to address these issues. It is the responsibility of all of you to share the information with people. It is the responsibility of elected officials to address these concerns. Being made aware of these concerns, they now have a responsibility, a legal, a moral, and ethical responsibility to share this information to address the concerns for the safety and the public health benefit of all Americans at the state level for the people in their states and at the national level for all Americans. We owe it to ourselves, we owe it to our loved ones, our friends, our families, our children, and our grandchildren. This is Dr. Richard Fleming, and today is Wednesday, the 27th of January, 2021.